Good afternoon, everyone, and Happy New Year. It's still until the end of this month we can say Happy New Year, so I'm saying Happy New Year to everyone. Hopefully 2021 will be better than 2020. And today we have said seminars in LCN series, and the subject is nanomechanics. So as usual, we have three talks, one from King's, one from Imperial, and one from UCL. And we have with us Giuliani from Imperial College London, who will be talking about in situ stable fracture tests. Then Sergi Garcia-Manyes from King's College will talk about mechanical stability of proteins. And finally, Bart Hoogenboom from UCL will be talking about nanomechanics in bacterial life and assassination. Very interesting titles. I'm looking forward to listen to them. And we go straight to recording of the talks.
Hello, my name is Dr. Giuliani. I'm going to talk about some of the interfacial testing techniques we've been developing to measure things like toughnesses of particularly interfaces in ceramics. So within material science, the mechanical properties interfaces are massively important, but within ceramics it's particularly critical, as it's one of the few mechanisms we have to actually increase and control the toughness of the material. So interface engineering has been a key technique within the structural ceramics community for a while. And this is just an example of two silicon carbide ceramics that have been etched, but they've been engineered to have very similar grain sizes, very similar grain aspect ratios. The only thing that's really different about them is the interfacial chemistry. So in one case, the liquid phase center material has a glassy phase, and the solid state material has a mixture of carbon and boron. What you see is that if you measure the long crack toughness, you start to see that the liquid phase center material has twice the properties of the solid state material. And this has just come about from the changing the chemistry of that interface. So a number of people have looked at ways to try and measure these interfacial properties. So we're not the first people to do that. There's some people that look at single cantilever beam typed experiments where you have a beam, you have a small notch. Some clamped beam where you then stick your notch underneath, and some flat punch type experiments where you essentially put pressure onto this sort of shape of material. You applied then a bending moment to that notch, and you then drive a crack down that pillar. And these have all had different levels of success to measure interfacial properties. They do all suffer from one slight issue, is that they're all dependent on a crack initiation effect, right? So how much stress you need to get a crack to grow at these points here, here, and here, which means how you make that notch is quite important. Some of them also are carried out, most of them are carried out within a nanoindenter, some are carried out within a normal lab environment. So things like moisture, particularly in ceramics, can actually play a role, can attack this crack tip, and then you can have problems, an actual chemical attack at that crack tip. We've gone for a slightly different route, where we've looked at doing the testing in situ within an SEM, and more importantly, we've come for a geometry where you should get stable fracture. So what we do is we have a sliver of material that we make in the fib, so it's about 2 microns wide by about 6 microns deep, and we drive a wedge into that material. In the same way as you might try and split a piece of wood, as you drive the wedge down, the crack should propagate in a stable manner. And here we can see the test. So this is our wedge coming down from the top, being driven down here. And then once we reach a critical load, we then start to propagate a crack. But what we're able to do here is we're able to propagate a crack under a stable condition. So we don't actually need to bother and use data from the initiation event. You can see that we can then hold this steady. This whole experiment here is about 20 minutes worth of work that's been sped up. Then we can unload it and you actually see the crack zips back up again. So that's fine. We can show that we can grow a stable crack in a crystalline material. How do we actually get a fracture energy out of it? Well, the basic idea is we treat either of these arms as a beam under bending conditions, and we balance the energy that's released as the crack grows with the energy that's released as the beam gets longer. There are a few things you need to take into account. These normally, if you consider the standard Euler-Bernoulli beam, you'd be looking for an aspect ratio of about 1 to 10. Here we vary from, I don't know, 1 to 4 up to maybe 1 to 6 or 7. So what we actually do is we have to take into account a rotation that can go around this root, and also we need to take into account the thickness of the beam, so we need to take into account the shear stresses. Once you do that, you can have a very simple relationship that gives you a fracture energy, which is related to the elastic modulus of your beams and some geometrical effects, and then we track how far these beams have moved, how far they've deflected, just with some image tracking scripts that we've written, and we also track how far the crack has advanced. You put that together, and what you're actually able to plot out is a fracture energy against crack length. And what you'd hope to see, at least for a single crystal, is that this is essentially uniform with crack length, because what you're doing is you're not expecting to see any other dissipation mechanisms. It's just how you break this material as you drive the wedge in. We can repeat this for a number of samples, and we do find that our small crack lengths can often be susceptible to initiation effects. We can see this here. Crack starts initially to go off sideways and then moves back centrally into the pillar, but otherwise we get quite good agreement at what we would class as long cracks. Compared to any kind of large-scale fracture testing measurement, 4 or 5 microns, it's tiny, but for us these are classed as quite long cracks. What it allows us also to do is build up a large number of data points. So what's interesting is that as we're actually splitting this material, we wanted to see how well our data agreed with the surface energy that would be calculated for silicon carbide. So we worked with James Commode's group in the University of Warwick, who looked at 2 different mechanisms to calculate the surface energy for the C-plane of silicon carbide and the various terminations that might be there. And you can see we get a number around about somewhere between 7 and 8 joules per meter squared for the 2 gamma value. Interestingly, if we compare this then, so here we have previous literature measurements for the fracture energy of single crystal silicon carbide at large scales, and then we have our experimental measurements here with the error bars, and then we have the first principles calculations from James' group. You actually see you have quite good agreement between our data and the first principles calculations, at least far better than you would from the large scale testing. So what it seems here is we're actually getting quite a good measurement of the surface energy of a single crystal of silicon carbide. That's quite interesting just from a point that goes right back to where you would start off thinking about fracture mechanics and how Griffith thought about fracture mechanics. It actually supports that initial idea, but it also means that we should be sensitive to changes in chemistry of any interfaces we then go on to measure. So all we're really doing here is we're measuring the energy to break those bonds down that plane, and we don't have any other dissipation mechanisms. We've also carried out these experiments on diamond. This is a far harder material to test, simply because it stores a vast amount of energy and it's very difficult to get a sharp notch, but we do get a number of frames out before our crack veers off laterally. And what gave us a certain amount of confidence is we're also getting good agreement from what people would calculate for the surface energy as we would measure in our experiments, right? So we now have a couple of data points from quite different materials that are giving similar answers. But really we haven't been that interested in measuring single crystals, we actually want to measure interfaces. So how we started off was to measure bicrystals, which we made by taking silicon carbide and spin coating a thin layer of TEOS on it, and then diffusion bonding them together to get a thin glassy layer between our two single crystals. These were then tested in the same way, and you see here we have our single crystal data for 4 samples, and then here we have our interface data for 3 samples. So 2 things to take from this. One, that it's far lower, which is as we would expect from the idea of having a crack deflecting interface in a ceramic. It's also around about the value you would expect for a glass. And interestingly, the data is actually very well behaved, and I think this is because the crack's actually drawn into that interface and is very well defined as it moves down through the crystal. So can we now apply this to more interesting interfaces? We've also looked at metal ceramics. So this is where you might expect to have some actual secondary energy absorption mechanism, right? So we've made, again, bicrystals by taking sapphire. We've sputtered on thin layers of niobium. We've simply taken the system because it's a system that's been well known to be able to diffusion bond fairly easily. We then put these 2 wafers back to back, temperature and pressure under vacuum, and then we form a bond where we have a sapphire sandwich essentially with a thin niobium layer in between. And you can see that on the right here we have the SEM layer, and these were ranging between 30 nanometers up to 200 nanometers, and this is a TEM image. You can see that we don't have any porosity, we have some grain structure, right? So we have grains 500 nanometers, a micron in width, but otherwise it's a nicely well-behaved layer. So we can carry out similar experiments as we did before. And if we look at a 30 nanometer interlayer layer, it's quite noisy data, it jumps around, but actually if we take a line through this, it's pretty much giving us the kind of area we would expect for the surface energy of a niobium to sapphire interface has been calculated in the literature. So it's noisy, but it's essentially flat. If, however, we compare this with now a 60 nanometer interlayer, so we've made this interlayer thicker, hopefully you can start to coming down the side here. We now start to put some load on it. It opens, but then we get to a point where it pauses, and you can actually start to see plastic deformation in this region here, the layers being pulled out, and correspondingly we see a massive increase in the energy. So previously we were thinking of energies around here, 5, 10 joules per meter squared, and now we start to see we go up to 100, 200 joules per meter squared. Now there's some variability in this, actually where this peak is, due to exactly where you say the crack tip is, but really we can see why we're getting a far greater energy absorption here, because we're getting this plasticity around the crack tip. And we can see that this is a couple of tests, so the one we've previously seen here in orange, another one going up here in blue, and then the long crack data, the thin layer previously. And what you can see is that actually they both end at a similar point where the crack goes down really nicely. But if we look at the peaks, you can see at these points where we're really drawing this material apart, and this is where we're actually starting to get the plastic deformation. We can see where this is larger and wider. But as I said, once you get to a longer crack and you're free from these areas where we're getting plasticity, the crack then runs smoothly, and we're getting back down to very similar values to the degree. So in summary, we've shown that it's possible to generate stable fracture in silicon carbide and diamond at the microscale using a double cantilever geometry. The major advantage this gives us is it allows crack initiation effects to be ignored. So any damage from milling or sample preparation can essentially be ignored because you're always fracturing virgin material under a stable geometry. This also gives us far more data points than you might expect from a standard fracture test. We might get 80 or 100 data points rather than one you'd get from a normal load control test. What's also interesting is if we measure fracture toughness with crack length, we can actually see the effect of microstructural features on the fracture energy. So we can pick up where we have debonding or where we have plastic deformation, and we can start to understand how these contribute to larger scale effects. And so, as I said, finally, we want to then use this to deconvolute more complex fracture problems. We also applied this to things like situations where you have chemical attack and understanding how that changes the crack growth rate, or growth energy, I should say. And with that, I will finish.
Good afternoon, everyone. My name is Sergi Garcia-Manyes, and I work at King's College London and at the Francis Crick Institute. So today I'm going to talk to you about some of the recent work from my lab, which I entitled "The Mechanical Stability of Proteins Regulates the Translocation Rate into the Cell Nucleus." My lab focuses on single protein mechanics and its direct implications on mechanobiology. And this exciting and rapidly developed field of mechanobiology, as its name implies, studies the effect of mechanical perturbations on an increasingly large number of biological processes, including cellular growth, differentiation or proliferation, or motility. And most of the work in the field has been focused on the cellular and tissue scale, and perhaps our understanding on the molecular origin of many of these mechanical processes is a bit less explored, mostly due to the lack of suitable technology that measures forces across different force and length scales. To rationalize the origin of the mechanical resistance of individual biomolecules, we need to investigate the behavior of the constituent chemical bonds when exposed to mechanical forces. Depending on the precise interaction that needs to be mechanically disrupted to induce a conformational change, the required forces will vary over wide ranges. For example, peeling the two DNA strands apart requires forces of only a few piconewtons, which is the consequence of breaking a few hydrogen bonds, which is this region over here. Proteins have mechanical stabilities that span from 5 or 10 piconewtons all the way to 300 or 400 piconewtons, and it's a consequence of breaking a large number of hydrogen bonds at once. And finally, a mechanically rigid covalent bond, such as a disulfide bond, requires forces of a few nanonewtons to break. And hence, given the large span of forces, a single technique is normally not enough, and the combination of three complementary different techniques is able to measure forces over a wide range. In particular, the three main techniques are the optical tweezers, the magnetic tweezers, and the AFM. My lab works with AFM, and most recently has also started working with magnetic tweezers. Just as a recap, the AFM is based on a sharp cantilever with a nanometer tip on its end. And an individual polyprotein can be tethered between the cantilever tip and a gold substrate, and the forces involved can be measured by the simple Hooke's law. Single molecule AFM can work in force extension mode by pulling the individual biomolecule at a constant velocity, measuring the protein's force as a function of distance, or in the force clamp regime, measuring the protein extension as a function of time under a constant force, which is achieved by employing an external PID electronic feedback. This is a sketch of how the experiment works, where an individual polyprotein is tethered between the gold substrate and the AFM tip. We normally work with polyproteins, which are formed by concatenating cDNA, which provide an ambiguous molecular fingerprints. A large body of literature on the single molecule behavior of proteins with AFM has focused on muscle proteins, and in particular on titin, which is the larger protein of our bodies and responsible for the passive elasticity of muscle. Titin is formed by a large number of Ig and fibronectin-like domains arranged in tandem, intercalated by protein regions that are intrinsically unfolded and function as entropic springs, which is this scheme over here. In the most typical approach, we can identify one such protein, so one such domain, such as the classical I27 domain, and build individual polyproteins. In the most classical AFM experiment, when proteins are pulled at a constant velocity, they unfold in a sawtooth pattern, where each particular force peak corresponds to unfolding one of the domains within the polyprotein chain. Each protein can be unambiguously characterized by the force at which the unfolding occurs, and also by the distance between two consecutive peaks, which is called what we call the contour length. Two of the main conclusions that we reach, or that were reached already at the beginning of the field, was the demonstration that polyproteins, when they are formed with different domains of different mechanical stabilities, unfold following a hierarchy in the mechanical stability, which means that when we pull on a polyprotein, the mechanically labile modules unfold first, which will be this one here, and then the mechanically rigid ones unfold later, irrespective of their position within the polyprotein chain. And an extreme case occurs when an intrinsically unfolded protein is intercalated, such as the PVK region of titin, is intercalated within the polyprotein chain. In this case, when the polyprotein is stretched under force, such intrinsically unfolded protein, devoid of any mechanical stability, would unfold first, which means that it doesn't oppose any resistance to the pulling force, followed by the unfolding of the mechanically resistant modules, which will be these ones here, occurring at higher forces. So all this general information on the basic features of the single molecule nanomechanical experiments would be somehow useful to framework the work that I'm going to be presenting today. My lab works on protein mechanics. However, I have recently become interested, and I would say almost obsessed, by trying to understand how the mechanical properties of proteins that we measure at the single molecule level can be scaled up and have knock-on effects at the cellular level. And for this, we design and develop different techniques able to measure forces at different length scales, ranging from individual proteins all the way to cells and even beyond. In particular, a central topic in our and many other labs is to understand how cells are able to sense extracellular mechanical signals and transduce these mechanical forces deep inside the cell, will be to here, to ultimately reach the nucleus and regulate gene expression. And with this very complex picture, an element that remains largely unknown is whether the passage of mechanically activated molecules through the nuclear pore complex, which is the main gateway in and out of the nucleus, is also mechanical in origin. So our motivation was to test whether, by controlling the behavior of individual molecules passing through the nuclear pore complex, we can induce functional changes at the cellular level. So one of the ways to propagate mechanical stimuli from the cytoplasm, which is here, into the nucleus, is through the activation or translocation of mechanosensitive transcription factors, which typically lie on the cytoplasm and, upon mechanical stimulation, are able to shuttle into the nucleus through the nuclear pore complex to find their target genes. And in collaboration with a group of Pere Roca in Barcelona, we demonstrated that by applying mechanical forces directly onto the nuclear envelope, we could mechanically deform the nuclear pore complex and regulate the kinetics of nuclear translocation of a particular transcription factor, which was called YAP. But we soon realized that this was only part of the... Because thanks to recent developments in cryo-EM, we have now very good structures of the nuclear pore complex. And despite the hole being quite large, which is about 40 nanometers in diameter, the nuclear pore complex is filled with this dense mesh of proteins called NUPs that are rapidly fluctuating intrinsically disordered proteins and create a physical barrier to translocation. The kinetics of passage to the nuclear pore complex is known to depend on the size of the cargo, and very recently has also been discovered to depend on the chemistry of the exposed amino acids, in the direction that hydrophobic residues have on average a much faster translocation rate than hydrophilic ones. And that made us think, because a classical globular protein has the hydrophobic amino acids hidden inside the protein and exposes the hydrophilic residues to the surface. So a potential way to rapidly expose the cryptic hydrophobic residues rapidly outside to expose them to the solvent is obviously through unfolding. And this, together with the observations from Carlos Bustamante and Bob Sauer, showing that in the Clipex proteasome machinery, proteins need to unfold to go through the pore, although this pore is admittedly much narrower in the nuclear pore complex before being chopped into individual amino acids, and similar experiments using nanopores in the group of Hay and Bailey in Oxford and others show that to go through this kind of hemolyzing pore that Hagen uses, proteins need to unfold first. So these are, to combine, all these experiments triggered us to conjecture that protein unfolding might accelerate the passage of proteins through the nuclear pore complex. To test this hypothesis, we chose a model protein transcription factor, which is called MRTFA. MRTFA is normally cytoplasmic and bound to G-actin. But upon mechanical stimulation, such as the presence of Rho GTPases, F-actin starts polymerizing, which makes the G-actin dissociate from the complex with MRTFA, and which reveals the previously cryptic NLS sequence that guides MRTFA into the nucleus. And in particular, as a transcription factor, MRTFA activates genes involved in cell adhesion, in cell migration, and actylin self. And one advantage of choosing MRTFA is that it's very well known at the biochemical level how to induce its translocation into the nucleus using serum stimulation. So when cells are serum starved, such as in these conditions here, MRTFA lies in the cytoplasm. But when stimulated, then MRTFA shuttles into the nucleus in a very quantitative way. So now, by tagging MRTFA with the GFP, we can follow with a confocal the dynamics of nuclear translocation upon serum stimulation in real time. In this case, these are cancer U2S cells, and measure the average dynamics for an ensemble of cells. You can see how, upon serum stimulation, the transcription factor shuttles into the nucleus. We can measure in each particular case the ratio between cytoplasmic and nuclear transcription factor. We can do that for an ensemble of cells and measure the average kinetics. And even after serum stimulation, there is always an equilibrium between nuclear and cytoplasmic MRTFA. And by fitting the average dynamics with a simple equilibrium kinetics, we can extract the import and export rate constants and the nuclear accumulation of MRTFA in equilibrium. And then the main question arises, really. Are these import and export rate constants affected by the mechanical properties of the translocating proteins? In other words, does mechanical unfolding of proteins accelerate their passage across the nuclear pore complex? If that were true, then a mechanically weak protein would unfold easier and translocate faster than a mechanically rigid one. To test this hypothesis, we engineered a protein chimera whereby the tagged GFP labeled MRTFA with a protein marker of known mechanical stability. And then we independently studied the mechanical properties of this mechanical reporter using single molecule force spectroscopy AFM, which we know how to do. Right? This is a video, a recap of how the AFM would work on this independent protein marker. As first candidates, we use different domains of titin, which is very well studied, and we know that the mechanical stability of the different IG titin domains increases on average from the N to the C termini. So we pick three different IG domains, the I1 in the proximal region, the I27 in the distal IG domain, and the I32 module, which is placed further down into the distal band. And then we constructed polyproteins of each individual domain, and we measure the mechanical stabilities using the AFM. And as expected, the I1 unfolds at 140 piconewtons, the I27 at 200 piconewtons, and the I32 at 270 piconewtons. So there's a pretty large spread of forces here. And then we fused our BFP labeled MRTFA with each of the IG domains, and we measured in each case the kinetics of nuclear translocation upon serum stimulation. And from the images, you could already see that for the I1, which is mechanically labile, there's a much larger fraction of nuclear accumulation compared to the mechanically resistant I27 or I32. In fact, we were really pleased at seeing that the average traces follow the hierarchy of mechanical stability. When the MRTFA was stuck with the mechanically fragile I1, we observed a much faster nuclear translocation rate in overall nuclear accumulation than for the mechanically stronger I27 and I32. And fitting the average dynamics trajectories to the equilibrium kinetics shows that nuclear import is exponentially dependent on the unfolding forces that we measure with the AFM, whereas the nuclear export is pretty much independent on the unfolding force. However, the different nitrogen domains of Titan, while having the same fall, they all have slightly different sizes and sequences. And strictly speaking, the definitive experiment should involve the modulation of the mechanical stability of a protein with exactly the same fold and exactly the same sequence. So to this end, we went back to the classical I27 protein, and we based ourselves on the experiments that Julio Fernandez did a few years ago, where they found that a single point mutation within the structure of the I27 protein changed its mechanical stability. However, in these cases, the changes in force from one mutant to the other one were very small, of about 10 to 15 piconewtons in some cases. So we repeated experiments and fused each mutant to the GFP labeled MRTFA transcription factor. And once again, the average in pore kinetics followed the mechanical stabilities measures in the AFM, with the import rates being still exponentially dependent on the unfolding forces that Julio's lab measured with the AFM, and the export rates being fairly independent. And as a final control, coming from the single molecule field, I was a bit scared with working with cells, and felt a bit uneasy about the variability that one observes from cell to cell. So to make sure that the average traces were truly representative, I asked my postdoc Elvira, and I think she hated me for this at that time, to do an experiment whereby we transfected the same cell with two different MRTFA now constructs at the same time. So one in blue, containing the MRTFA tagged with the high mechanical stability I27 wild type, and the second one in yellow, fused with a mechanically labile mutant of lower mechanical stability. And then inside the same cell, we compare the rate of nuclear accumulation in a sort of competition assay. And already from the images, you can see that at any given time, the yellow, which is the less mechanically stable mutant, shows a much higher level of nuclear accumulation than the mechanically stronger, so wild-type protein form. And this is in fact statistically confirming the average traces on the average curves, showing different levels of nuclear accumulation. So overall, that put all of my concerns at rest. Okay, so now the final question is, can we modulate gene expression and cell function by changing the mechanical properties of the translocating proteins? So we first measured the mRNA levels of myosin light chain, actin, and SRF, which are known to be upregulated by MRTFA. And we found that those cells expressing mechanically stable MRTFA had significantly lower levels of mRNA than those expressing MRTFA with lower mechanical stability. And then we looked at cell migration. We conducted these cell scratching experiments whereby we measured for each stable cell line the efficiency in closing the wound for cells expressing a mechanically rigid and a mechanically labile MRTFA. And found that those cells expressing the low mechanical stability B13 mutant closed the wound much faster than those cells expressing the MRTFA tagged with the mechanically rigid I27 wild type. And similarly, we measured the motility of individual MDA MB231 cells, which are a breast cancer cell line that is highly metastatic. And by tracking the motility of each cell over time, we observed that cells transfected with mechanically rigid MRTFA move significantly slower than those transfected with the mechanically labile MRTFA. So coming to the conclusions, we believe that we can say now that the mechanical stability of proteins seems to be an additional factor in addition to size and to chemical composition to control the dynamics of nuclear imports to the nuclear pore complex, and that this has knock-on effects at the cellular level. So I'm going to wrap up here, but before finishing, I'd like to thank all my lab, and in particular the two key players in this project, Elvira Infante, a cell biologist, and Andrew Stanner, a physicist that work together in this interdisciplinary project. And of course, all the funders that are supporting the research in my lab. Thank you all for your attention, and I'm happy to take any questions.
Since I presume that several of you might be juggling attendance to this seminar with, say, homeschooling of your kids, I thought I'd start with the question my 7-year-old son Louis asked me this morning, which was, so why is Cinderella not good at football? And I thought, well, maybe because she has only one shoe. But that's wrong. The correct answer is because she's always running away from the ball. Now that's as far as my entertainment for primary school age goes. In the next 10 to 15 minutes, I'll try to entertain you with not so much ready research results, but things we are working on, leading to still unanswered questions that I hope will intrigue and interest you. The focus of this research is the gram-negative bacterial cell envelope. This is an envelope that surrounds all gram-negative bacteria, like E. coli. And it's a very important envelope because it protects bacteria against antibiotics, against proteins and peptides from the immune system, against harsh environments, and it is in itself also a target for several antibiotics. Now, it's also scientifically interesting because it consists as it is, unlike say eukaryotic membranes, it's a composite cell envelope. So as in eukaryotic cell membranes, you have a lipid bilayer which provides what we call the inner membrane. Then surrounding the inner membrane we have what we call peptidoglycan layer, also called cell wall, that consists of sugary chains that provide a lot of stiffness to the cell envelope. And then on the outside we have what we call the outer membrane, which is a layer consisting of lipids on the inner leaflet and so-called LPS molecules on the outer, and relatively densely populated by porine molecules. We study this, we and actually other people study this cell envelope with atomic force microscopy. And briefly what we do is we look at bacteria on the surface, we scan the contours of the surface with an AFM tip, and then basically translate that in an image. We can do so by tracing the bacteria on direct contours, and that's all, or we can do so by actually modulating the height of our probe above the bacteria, and that way we continuously record force curves, where we record the force on the AFM cantilever as measured by the deflection as a function of the distance above the surface. And so that means you can get curves where you have a contact point and then you have an indentation, and the steepness of this curve here allows you to infer information about mechanical properties of the sample under study. Now others, in this case Jamie Hobbs' lab in Sheffield, have used AFM to image the cell wall, so these peptidoglycan layers on isolated cell envelopes, where you see that they consist of many chains which are arranged in a rather disorderly way, and actually how this is forming the cell wall, how it is providing mechanical stiffness are all questions that we still need to answer. In another example of AFM work on bacteria, in this case by K.C. Wang's lab in California, they measured the stiffness of various bacteria by AFM, and then looked how, compared to the wild type stiffness that you see over here, that's the normalization value here, 1, you see various mutations like OMP-A, which is a protein that provides linkage between the cell wall and the outer membrane, and LLLP, which does essentially the same, led to a softening of the layer, and this combined with various other experiments led them to conclude that in this composite cell envelope you have the cell wall might provide stiffness, but actually it's the outer membrane which also provides a lot of stiffness. In basically defining how bacteria responds to stress, how bacteria resists forces in nature. Now, my own lab's entry to bacteria occurred via a whole body of work that we've done on pore-forming proteins, which are proteins that are secreted, whether this is by bacteria or by the immune system, in solution. They exist as monomers in solution, they land on the membrane, they assemble there, and via various pathways they ultimately form arc or ring-shaped pores in the membrane, where the diameter here can be 10, 20, sometimes even 30 nanometers, typically to lyse target cells. Now, for today, I'll focus on the membrane attack complex, which is a complex consisting of complement proteins. Complement is a system of proteins in our blood. And what this membrane attack complex does is it punches holes in bacteria, and particularly in gram-negative bacteria. And if gram-negative bacteria enter the bloodstream, the membrane attack complex is basically punching holes in these bacteria and makes them and causes them to die. The membrane attack complex is formed of multiple proteins. You have a sort of initiator complex, which is consisting of C5, C6, C7, C8, and then we have multiple copies of C9 that completes this ring. Now, this looks all nice, and we have studied this quite extensively. We originally studied this on supported lipid bilayers as model bacterial membranes, where the whole idea was we fuse vesicles on the membrane such that we get a completely covering lipid bilayer. We inject the proteins and then watch the pore formation in real time. You can see this for the membrane attack complex. We actually have these types of pores. We see this stalk protruding, so we can resolve them really well. And if we scan a bit faster at the expense of some resolution, we can take such a model membrane for an E. coli membrane and subsequently add the different components. So we can add C5BE6, we can add C7, we can add C8, and you start to resolve a few blobs here and there on the image, and then we can add C9. And what you can then see is that relatively rapidly pores appear, and as soon as a pore starts to appear, it completes very rapidly. And images like this allow us to identify kinetic bottlenecks in this important process in immune killing of bacteria. But this is of course on a model membrane, and that doesn't tell us necessarily what happens if we look at real bacteria. This was work done by Ed Parsons, then a postdoc in my lab. And more recently, with help of others, Georgina Ben, currently a PhD student in my lab, she worked out and established protocols where we can really robustly and repeatedly adsorb E. coli on a glass slide, keep it in solution, image it by AFM, and then look at the surface of these bacteria. And actually we resolve the outer membrane at remarkable detail, and this detail is something we are still trying to understand better in terms of the organization and order that exists for the proteins in this outer membrane. One of the nice things we can do there is that we can monitor at the same time by AFM the surface of the bacteria, as you see it over here and in this image over here at the scale of nanometers, while also using an inverted optical microscope locate the bacteria. So this is now at a much lower resolution, but we can clearly see the bacteria in a bright field image as these cigar shapes. And when we have a nucleic acid stain in solution, we can also check whether these bacteria are alive or dead. And so the ones which fluoresce red here in this image are the ones which are dead. So we can do AFM images on both living and dead bacteria, and we can verify actually whether they are alive or whether they're dead or whether they're agonizing and dying. Talking about bacteria dying, it's been known for over a century that serum can kill bacteria, and we now also know that basically one of the ways of doing that is by the formation of the membrane attack complex, punching holes in the bacteria. Now that sounds all very simple, but as often in biology the reality is a bit more complex, in part because, as I've told you, the bacterial cell envelope is a composite envelope, and you have to simply punch holes through different layers. As a consequence, the basic question, how does the membrane attack complex kill bacteria, is still unanswered. And I'm afraid I'm not going to give you an answer in the remainder of this talk, but I'm going to give you some hints that we have that mechanics of the cell envelope might play a role there. As imaged by AFM on live bacteria, the membrane attack complex looks as follows. We can start off with the bacteria displayed as a sort of cigar. This is E. coli again, as before. We can zoom in and resolve actually quite some detail about the outer membrane, which is at the moment somewhat hard to identify still, but that's something for a different talk. And when we expose them to membrane attack complex, we see a bit more protrusions on the surface of the bacteria, and if we zoom in, we can resolve here these pores at a few nanometers resolution, clearly targeting and porating the outer membrane. To learn more how things develop when you look below the outer membrane, we can use fluorescence methods, and these are all experiments done in Suzanne Royaker's lab in Utrecht in the Netherlands, with whom we collaborate on this topic. And what is done there is that we can use bacteria that have M-cherry, a red fluorescence marker, in the periplasmic space, in the space between the inner membrane and the outer membrane. So if the outer membrane is compromised, this would leak out and gives us a measure of outer membrane perforation. Now, inner membrane perforation can also be measured by GFP, which is in the cytoplasm, so inside the cell, which we don't really use that much. But what we can also do is, as shown before, we can use a nucleic acid dye, which is in the solution, and if both the outer membrane and the inner membrane are compromised, it can go in through the whole cell envelope and provide us a marker of the whole, how the cell envelope as a whole is compromised. Now, this gives data like shown here, where we have on the left axis, we have the M-cherry signal. Here for various controls remains high, so normalized here on a buffer control. So that means that the fluorescence is still in the bacteria, in the space between the inner and the outer membrane. And when you then expose the bacteria to the membrane attack complex, you see that this drops down as a marker of outer membrane perforation. At the same time, we can monitor, here plotted on the right axis, the staining of bacteria by nucleic acid stain, which you see is pretty much 0 for the control, and where you see that the inner membrane is compromised, where the bacteria stain for cytox once the bacteria are exposed to the membrane attack complex. Is basically what you have in this column here. So you see the M-cherry signal going down, means the outer membrane is perforated, and you see the cytox signal going up, which means the inner membrane is perforated. Now, the interesting thing we can do there is we can look at what happens if we only expose the bacteria to C5, C6, C7 and C8. So you only have this part, and that's the column over here where you see that the outer membrane is not significantly compromised, maybe a tiny little bit, and there might be some leakage in the inner membrane. And then what we can do is we can expose the bacteria to C5, C6, C8. We know the outer membrane is not compromised or hardly compromised at all, then wash extensively and then expose them to C9. The idea is that that way you can form this precursor complex only on the outer membrane, remove all precursors except from the one that are already bound to the outer membrane, add C9, form pores in the outer membrane. But at that point we can't form pores in the inner membrane anymore because the precursor is lacking. And when you do that, you see that basically the bacteria are compromised in the same way as when we just expose the bacteria to the membrane attack complex as normal. You see that the outer membrane is compromised and the inner membrane is compromised, shown here by the drop of the M-cherry signal compared to the control and the increase of the cytox signal, the nucleic acid stain in the bacteria. So this tells us that the pores, the poration of the inner membrane or the permeabilization of the inner membrane is not of the same nature as that of the outer membrane. So that brings us to the question, so how do we get across this composite membrane? And so we have the outer membrane, the peptidoglycan layer, the inner membrane. We have things which I haven't discussed here, which is about how exactly enzymes at the bacterial surface facilitate membrane attack complex formation and insertion. But we get pores in the outer membrane that we can also see by AFM. We have extensive outer membrane damage, and indirectly this causes permeabilization of the inner membrane by a mechanism that we still don't know. Now, more recently, we have tried to look at this bacterial killing sort of in real time, where you see here the bacteria, E. coli is imaged by AFM, here with an arbitrary offset, 0 seconds. The pores as formed on the bacteria, so this is 0 seconds, half a minute later, we still have pores. And what we can also do is using our fluorescent staining, we can take fluorescent image where you see here the contour, the shadow of our AFM probe. We see here one bright spot which corresponds to one bug, which is dead, that's why it's bright. And there's another bug which is located over here, which we don't see because it's not stained yet, which is the one that is underneath our tip. No staining, so that bacteria is still alive. Now, if you wait a bit longer, we suddenly see we go from this type of AFM image where we can clearly resolve the membrane attack complexes on the surface to something which looks a complete mess. And in AFM talk, this means that the whole surface here is destabilized. We can't resolve it very, very well anymore. It is too wobbly for us to trace the surface reasonably. So we see a mechanical destabilization of the outer membrane. And then if we go back to our fluorescence staining, you see that we now have, at the position where we know the tip is, we have another bright spot which corresponds to the bacteria being now dead. So what we see here in the still preliminary data is that we have the mechanical destabilization of the outer membrane that coincides with the bacterial killing by the membrane attack complex. Now, of course, you can ask, what's the chicken, what's the egg here? Is the mechanical destabilization now the reason that the bacteria die? And that's one of our hypotheses. We extensively porate the outer membrane, leading to the destabilization of the cell envelope as a whole, leading to cell death. Or something which we cannot not exclude yet, could it be that the bacteria just die for another reason that we still don't know, and that causes the overall destabilization? So that's still what I think intriguing question that we're still trying to answer. Again, trying to explain the basic mechanisms by which our immune system can kill gram-negative bacteria in the bloodstream. And as I warned you in advance, we haven't got the answer there yet. Now, with that unanswered question, I would like to conclude, and I'll thank collaborators who have contributed to this in my lab in the past and in the present, named over here. I mentioned already Georgina for her recent work and Ed for his work on resolving membrane attack complex as it was formed on supported lipid bilayers. The work on supported lipid bilayers or model membranes is together with Dorian Bubeck at Imperial College. I mentioned Suzanne Royaker's lab in Utrecht and her collaborators with whom we do all the work on live bacteria. And for AFM technical and other surface probe techniques, we had assistance from Georg Wandner's lab at EPFL Lausanne and from Ralph Richter in Leeds. And finally, I'd like to make use of this opportunity to advertise for a BBSRC case PhD studentship that I'm currently recruiting for, not on this topic, but on another interesting topic. And for anyone who's interested in that, please let me know. I'll be happy to talk about that. Thank you for your attention.
Thank you very much, Finn, Bart, and Sergi. Very, very interesting talks, different, and all talks are very interesting. So unfortunately, we're running out of time, but we have a few minutes for questions. Who would like to start? So maybe I will start. So I have a question to Finn. So what is the smallest size of the object that you can study?
I think in terms of laterally, I mean, it depends if we think about the arms, you're probably around a few hundred nanometers width. And then you probably have a ratio in terms of how far you want the crack to be, a multiple of that. So probably of that range. We haven't tried smaller ones. In some respects, they have advantages. They're more flexible. But this has been the geometry that's kind of worked for the things we're doing, but you could shrink it a little. You probably couldn't get down to the tens of nanometer range, but hundreds should work.
I see. And in terms of you always showed your data in terms of energy required to form the crack. So what actually the force you applying to the tip to initiate?
The force isn't really that interesting in a sense, because it is taken up with a lot of friction and it changes as the crack propagates. It's of the order of a few millinewtons, probably. But although we measure it, it's not something we actually use in the analysis at all, because it's actually very difficult to interpret.
Yeah. Thank you. More questions?
Hi. Yeah, I have a question for Sergi. I wanted to know, why is the export rate for the proteins not changing with the mechanical stiffness of the unfolding process? Because I wasn't able to understand that.
Another question, actually. So the export doesn't change. I mean, the short answer is we do not know. The thing is that once the protein unfolds, it doesn't refold back into when it's back in the nucleus, and obviously there's always a if you want, of export and import, and it's very likely that the exportings don't care about the mechanical stability, whereas when you start, you have to unfold, and that's why the importings do, and that's what we are. That's our hypothesis at the moment. We're testing that, and this all suggests that it's going into that direction.
Okay, thank you.
So unfortunately, we ran out of time. Thank you very much to our speakers. Thank you very much to all participants. It was very, very interesting talks, and we are looking forward to see you next time in a month's time. So this LCN seminars will be every month, last Wednesday of the month. So see you in February. Thank you very much.